Okay, good morning, everybody. And we are going to carry on our discussion about animal nutrition. This is the slide that we finished off with last class, and we were talking about the processes of digestion. I just want to remind you that all of the enzymatic digestions that we're going to talk about, just remind yourselves about how these catabolic reactions, the breaking of molecules, actually takes place. They are by hydrolysis. That is, we actually take a water molecule, that's where the hydro part comes from. We take a water molecule and effectively add it in here, in this, break this bond by putting in water. Here you can see an OH group there, there's the other H there, and the dissolution of the big molecule to produce the two products. Okay, so we're going to watch now, we're going to discuss the actual structure of digestive systems. We're going to see how it is that, first of all, the first processes of digestion, chewing, etc., take place, how some of the different processes of digestion in different regions of the digestive system, how they are absorbed, okay? And before we do, let's think for a minute about the variety of digestive systems that are present in the animal kingdom. It depends what you're talking about. In the simplest of the animals, that is things like single-celled organisms, amoeba or whatever, and the other group of really simple, the basal animals that we talked about, the sponges, the porifera, there isn't really no digestive system. In fact, those the cells in those animals act to take in their food, usually by phagocytosis, that is by actively swallowing particles from outside. There are no true digestive processes that take place external to the cell. They take place internally. In the simple animals, such as the flatworms, the cnidaria, the hydra, and those things, there is a digestive system, but it is a simple digestive system. And there is just one opening to the opening which functions both to take in material and to void the waste. And the cavity that it leads to is called the gastrovascular cavity. It gets that name because first of all, it functions as like a stomach. The gastro part comes from. But everything that is digested extracellularly and circulates in that cavity and is then absorbed by the cells within the gastrovascular cavity. And they are absorbed not by phagocytosis as they were in the amoeba and the sponges, those sorts of things, but they're absorbed by diffusive processes. The similar sorts of processes are then elaborated on in the higher animals. But now in the higher animals, there is a complex digestive system. That means that it is complete. It has a mouth and it has an anus. It's a tube with two openings. The digestion is still extracellular throughout, but very often in these complete guts, different regions of that complete gut perform different functions and have different digestive processes that go on, and different absorptive processes as well. So that is called a complete gut, and that's typical for all of the rest of the animals, from relatively simple invertebrates all the way up, all the way through to the vertebrates, etc. And so when we look at our own digestive system, it's really typical of animals. What it consists of is an elaborate tube, a very elaborate tube. And the elaboration, the structure of that tube is related, of course, to the diet and to how the food needs to be digested and absorbed. In our own case, here we see the tube running all the way through, mouth to anus. But accessory to that tube are complex glands, which are going to contribute the digestive juices, digestive enzymes, for example. which then are passed into the tube in order to effect digestion. So when we look at the structure of our own digestive tract rather, the first part of our digestive tract is from the mouth on, right? So the first part is the oral cavity, and there are a set of glands accessory to the oral cavity, which are the salivary glands underneath here, and then up around on the sides of the face there. And the salivary glands actually produce a complex mixture. Saliva consists of mucus. Mucus is viscous, and it consists of water. It has salts in it. It has abundant substances called glycoproteins, which is what gives it its slightly gel-like consistency, and which is what makes it viscous. But the saliva also contains amylase. And so amylase is the very first of our digestive enzymes, and produced by the salivary glands. And it is brought to action by being mixed with food during the process of chewing. And chewing cuts up the food. And that's why you chew your food well. It is true, you should chew your food well, because you want to, first of all, cut the food into a small set of pieces as possible with as much surface area as possible, but also to mix the food with the saliva, both to form an easily swallowed collection of food, which is called the bolus, and also to mix the first of the digestive enzymes, the amylase, with the food. Amylase is responsible for breaking down starch. Starch is a complex mixture, a complex structure made entirely of glucose molecules, all polymerized together, and amylase will break that down. The tongue forms that bolus, and it also pushes the food to the back of the throat. And once it is at the back of the throat, we make a voluntary decision to swallow. Once that decision is made, once the food has passed a certain point over the back of the throat, involuntary processes take over. So the structure at the back of the throat is really important. All the vertebrates share this feature, that the digestive tract actually shares anatomy with the respiratory tract at the very top here. And this is the area called the pharynx. So the tube which leads into the digestive tract is the esophagus, and the tube that leads to the respiratory tract is the trachea. So it's really important, of course, that food does not go down the trachea. In fact, this is a really dangerous situation. It can be a lethal situation. There is an anatomical adaptation to ensure that this doesn't happen. As the food passes behind here, there is a reflex reaction which causes the epiglottis, which is this flap at the top here. It's to flap over and close off the trachea like that. As the food passes, as the bolus passes, this epiglottis should close to protect the respiratory tract. This can go wrong. You can, for example, especially if you breathe in, which causes the epiglottis to rise at the same time as you swallow it, and the whole system can get confused, and you can end up with food passing into the respiratory tract. If it does, it often gets trapped at the top of the voice box, of the larynx. And that's a very dangerous situation. That is a situation of choking. And that is why we should all learn the Heimlich maneuver. Usually there is air in the lungs still, even if you breathe out. If food is here and you manage to compress the lungs suddenly enough, you can blast that food back out and away from the top of the trachea. By the way, just the word of warning, if somebody is choking at it when you're with them, as long as they're coughing, they're fine. Even if they're coughing violently, they're fine. Don't just comfort them and calm them down. The universal sign of true choking is this. And people, all that they can do, if they can make a noise at all, it's a high-pitched whining sound. They're not gasping for air or anything. They clasp like this and make this panicked look. That's the time to get behind them and give them a good Heimlich to see that if you can save them. So once this food has passed this point here, reflex processes take over, and the esophagus. Your food doesn't just plummet down your esophagus. The esophagus undergoes a rhythmic contraction and relaxation called peristalsis, which actively moves the bolus of food down the esophagus. This is why, and if you don't believe me, you can try it out. Just don't choke yourselves. If you stand on your head, you can swallow quite easily. And it's because as long as you can get the food to the top of the esophagus, which is easy, actively swallow, and it was passed to the stomach by peristalsis, not by gravity. There are, throughout the digestive tract, there are sets of sphincters. A sphincter is a complex set of muscles which form a ring, which can be closed and opened. And they act, they're not really valves. You can think of them as gateways, really. And here is the first of those sphincters. It's at the top of the esophagus, and that is relaxed instinctively as the peristalsis starts. There is another at the entrance to the stomach. So throughout the digestive tract, these muscular sphincters regulate the passage of the food from one chamber to the next, often very carefully controlled by the autonomic nervous system, the automatic nervous system, which monitors what is happening during the digestive process to move food correctly through the different processes. Now, once the food arrives in the stomach is a very complex organ. It certainly is the initial kind of storage area, and food is stored in the stomach and is passed gradually into the rest of the digestive tract. But digestion, there are several digestive processes that take place inside the stomach. And the stomach is an active muscular organ. And it is also a secretory organ. It has glandular qualities to it. The muscular action is to mix up the food very actively. The bolus of food, they all get mixed up together. And they begin to break down because the fluids in the stomach are extremely acid. And that begins to denature proteins, for example. In addition, the gastric juice contains a very powerful enzyme called, and the most important, actually there are a couple, but the most important one is pepsin. And pepsin is a powerful protease. It breaks down proteins. Both the acid, which is hydrochloric acid, and the pepsin are produced by the cells in the walls of the stomach, as I'll show you in a minute. But they're extremely powerful, so the stomach cells themselves need to be protected. And there are two mechanisms whereby that takes place. I'll detail this in a minute. As the food is processed by the stomach, and it produces a substance called chyme, and the chyme is passed gradually into the small intestine. Again, there are sets of sphincters which govern the passage from the stomach then into the small intestine. Now, the wall of the stomach is quite complex, and the wall of the stomach is densely involuted like this into these are actually called crypts here, and they form glands with openings that open into the stomach cavity. And when we look at the cells of these crypts, we'll see that they consist of different kinds of cells. The first are called the chief cells. And these produce a precursor to the enzyme pepsin. It's called pepsinogen. These big cells here are called parietal cells, and they actively produce protons, H+, and chloride ions, which are accumulated in here inside the cavity of the crypt, and are then passed through to the stomach cavity. So if they are making H+ and Cl-, a solution of H+ and Cl-, what are they actually making? They're making hydrochloric acid, which is a powerful acid. And the pH of the stomach is actually around about 2. And that is enough to actually burn naked flesh. So how is it that the stomach is protected? This is the first protective mechanism for the stomach, is there are also embedded in here, there are mucus glands, which cover these cells with mucus, which protects them from the hydrochloric acid. The second way in which the stomach cells could be damaged is by the very powerful protease enzyme pepsin. But when pepsin is produced here in the folds of the stomach wall, it's actually produced in a precursor form, pepsinogen. Now you need to think about this, because if a cell is producing a very powerful enzyme, that enzyme is floating around inside the cytoplasm. It could digest the cell from the inside out. This is why the enzyme is produced in a precursor form, pepsinogen. It is released into the stomach wall, into the stomach cavity, and there the low pH activates this. It actually cleaves it, and it produces the active enzyme pepsin. So those are the two mechanisms whereby protection of stomach cells take place. Once the chyme has been adequately mixed and has been the processes of digestion by pepsin, etc., have taken place, it is passed into the small intestine. And the small intestine is the largest of all of the compartments of our digestive tract. In human beings, it is around about 22 feet long. And it's in the small intestine that most enzymatic hydrolysis takes place. There are a large number of very powerful enzymes which are released into the lumen of the small intestine, which complete the rest of the digestive process. So the first part of the small intestine is called the duodenum, and it links the stomach and the rest of the small intestine. There are a number of accessory glands which put digestive fluids into the small intestine. The first of these is the liver. The liver itself is a gland, as well as various other, having their numerous other functions. And one of its important secretions is bile. And bile is created from the breakdown of red blood cells. The hemoglobin in red blood cells is actually processed to produce bile, and it's accumulated in a bladder, which is the gallbladder, and is then passed in a duct to the small intestine. The other really important gland associated with the small intestine is the pancreas. And the pancreas makes numerous substances which are released into the lumen of the small intestine. The one really important one is bicarbonate. And bicarbonate neutralizes the acid from the stomach. So the pH of the food passing through, the chyme as it passes through, is first of all very low. And then once it encounters all of the bicarbonate from the pancreas, the pH is raised to neutralize it. But the pancreas also produces several other important digestive enzymes that we'll hear about. So here, the pancreas is kind of hidden away behind here. it will lie behind the stomach here. Here's the duodenum, and leading to the rest of the small intestine. I'll show you more about the structure here. Here's the duodenum here. This first part of the small intestine is called the jejunum, and the second part is called the ileum. There are some histological differences between them, but we won't worry about the differences between them. Most digestion is completed in the duodenum. Most absorption of nutrient takes place inside the small intestine. So the small intestine is especially adapted for this absorptive process. Even though it's long, it's 22 feet long, it still has to achieve the task of absorbing as much nutrient as the animal needs in a relatively short space of time. And the cells of the small intestine are adapted. They may absorb a lot of nutrient simply by diffusive processes, but there are some nutrients which they will actively take up. And once all of that uptake has taken place, you should know that there is a special circulatory adaptation. And this is that under normal circumstances, blood, which is in circulation in the body, always passes to the heart and then out to the rest of the body. But in certain circumstances, and the gut is one of them, there is a direct connection between organs which does not go through the heart first, and that is called a portal system. And there is a portal system between the gut and the liver, and that is called the hepatic portal system. And the hepatic portal system takes all of the absorbed nutrients straight to the liver for processing, and then on to the rest of the body. So here's the structure of the wall of the small intestine. The absorption has to be accomplished efficiently and in a relatively small space of time. To do so, the wall of the small intestine is adapted so that it has an enormous surface area. The first thing is that the wall is cast into folds, which you can see here. That or immediately increases the surface area of the small intestine. But if we look carefully at the folds, these folds of the intestinal wall, we'll see that they themselves are folded like this. They are folded into what are called the villi, V-I-L-L-I. So these are finger-like projections like this, and they have their own blood supply of artery and a vein, leading to arteries and veins here. And in addition, they have a branch of the lymphatic system here, which is called a lacteal, which leads to a lymph vessel there. This is all very important, and it's important to understand that if we're going to understand how the small intestine actually absorbs material. So the first thing to understand is that the villi themselves, the surface area of the villi is enormously increased because each of the cells that make up the wall of the villus here actually has what is called a brush border. It is folded into these microscopic folds called the microvilli, which coat the surface of the whole of the interior of the small intestine. And this means that the surface area is absolutely enormous as we go through the whole small intestine. And the nutrient absorption can be efficient. It's easy to understand how small things like the amino acids, individual carbohydrates, these sorts of things can be absorbed by diffusive processes. And they are simply taken across, passed into the hepatic portal, and then out to the liver, and then to the rest of the body. But the treatment of fat is a little bit different. Fats are quite difficult to deal with, of course, because they are insoluble, and they have to be actually broken down into soluble components. They are made up of soluble components, of relatively soluble components, but fats in themselves are very big molecules which are not soluble and not easy to deal with. But we take in quite a lot of fat, especially if we're omnivores or carnivores. We take in quite a lot of fat. And it has to be, somehow it has to be processed. This is the way it's done. This is the function of bile in the small intestine, is to act as an emulsifier. That is, it will take a large fat droplet and it will break up the fat droplet into minute droplets. And they're not soluble, but they are emulsified. I'll give you an example. Mayonnaise is actually emulsified fat. That is the kind of effect of emulsifying fat, of breaking up large fat globules into very small fat globules suspended in a liquid. And that is exactly what bile does. It breaks up the fat globules into small fat droplets. And those are then digested. And they are digested especially by a powerful set of lipases. A lipase is something that breaks down fat into its component parts. And the lipases are produced, by the way, by the pancreas. There are some lipases also produced by the walls of the small intestine. But their effect is to break up the fat droplets. Think back to biomolecules. The structure of fats was a glycerol molecule with 3 fatty acid tails attached to it. The lipases break those down and produce fatty acids. They free the fatty acid tails, and they produce a glycerol molecule with just 1 fatty acid tail attached to it. And those can be taken up by the cells of the intestine. And they are absorbed by the cells of the small intestine. And once inside the cytoplasm, they are actually reassembled into fats. They're reconstituted as triglycerides. And they are then mixed up with a very complex structure, very small structure called a chylomicron. Here's one here. Here's the fat on the inside. And then around the outside, it looks like cell membrane, a plasma membrane, excepting it's only got 1 sheath, but it is phospholipid. And this chylomicron is small enough that it can actually pass across the plasma membrane, and it is soluble. It is soluble because the outside here are the phosphate heads of the phospholipid, which are highly soluble in water. But this chylomicron does not pass into the bloodstream. Instead, let me just go back here and show you here. That's the importance of this lacteal here, this lymph vessel there. They pass into that lacteal, and they then pass into the lymphatic system, and then into the circulation, and then to the liver and into circulation. So the processing of fat is actually quite a complex process, largely attached to the fact that it is not soluble. So most of our digestion, everything, absorption takes place in the small intestine. Once all of that is done, what is left is passed into the large intestine. And the large intestine consists of the 4 parts. This is all colon. This U shape here is the colon. And it consists of the ascending colon. The small intestine enters here, again through a sphincter. That's the ascending. This is the transverse, and this is the descending colon. The last part here is the rectum. And most of the waste food is stored here and then passed into the rectum immediately before defecation, before it is voided. So this colon, you may wonder why it's a large structure. and it must have functions. It has several functions. The first is that it absorbs a tremendous amount of water and consolidates the waste into the feces. The second thing that is important about it is that it actually contains the bulk of our microbiome, of our gut microbiome. The small intestine, even the stomach, also have an associated gut microbiome, but the colon has by far the most bacteria associated with it, and they are very important. They contribute all sorts of things to us. They contribute some nutrients like fatty acids and things like that, but they also produce vitamins, as I've told you before, and perform all sorts of other functions as well. So this colon here, in us, in actual fact, its function is relatively simple compared to the kinds of functions that we might see in other animals like herbivores. The food, once it's is compacted, passed into the rectum, it's voided in defecation. And again, of course, that is controlled by a set of sphincters. Okay, so use this diagram here to see how and where different kinds of biomolecules are actually processed and absorbed. Carbohydrates, their digestion begins in the mouth, and it continues in the small intestine. There are sets of pancreatic amylases apart from the amylase produced by the salivary glands, and they are broken down into disaccharides usually and absorbed, and then further digested to monosaccharides and absorbed in the small intestine. Protein digestion begins in the stomach with pepsin, but there are many different enzymes, proteases, which the most important ones produced by the pancreas are trypsin and chymotrypsin. But there are also some others as well. There's another one called the carboxypeptidases, which is responsible for breaking down small polypeptides, which result from this digestion. And then there are even more in the small intestine, and absorption of individual dipeptides and amino acids takes place in the small intestine. We can even digest nucleic acids. And of course, the fat digestion I've told you about, that is the responsibility of small intestine. Okay, so I've already hinted that we can see adaptations of this structure. Ours is quite a basic digestive system. We can see elaborations and adaptations of that digestive system, which depend on the nutrition, the lifestyle of the animal concerned. Remember, we talked about herbivores, carnivores, and then omnivores. Each of those groups is going to show special adaptations to their diet. And the very first adaptation is dental, where there are important adaptations of the jaws and of the teeth. The second is adaptations of the structure of the digestive system itself. And the third is really important, and that is the different organisms, depending on whether they're herbivores, carnivores, et cetera, they have different relationships with their gut bacteria, which are tremendously important. So first of all, looking at teeth, a biologist can usually immediately know what group an animal falls into by looking at their teeth, because the teeth actually are really distinctly adapted for a particular diet. And this is particularly true of the mammals, because we have differentiated teeth. The reptiles do not. The reptiles, fish, those, and everything else, they don't have teeth which are modified into groups with separate function. All of their teeth are alike. In the carnivores, the first thing that we notice in the carnivores is they have these big teeth, which are the carnivorous teeth. These at the front here are called the incisors, And in carnivores, they tend to be somewhat less important than these carnivorous, these canines here. But the real hallmark of the carnivores are these back teeth here, because the back teeth form scissor-like slicing devices, and they are called the carnassials. And that is the defining feature of the carnivore skull. You may even have some carnivores with relatively small canines. Some of them have enormous canines, but they have carnassials. And those are literally, they act like two knives slicing. Of course, the carnivore depends on meat for its diet, and that has to be cut into small pieces before it's swallowed and digested. Herbivores are adapted to actually grind their food really finely. So they have no canines, they have no carnassial teeth, but their molars and premolar teeth are specially adapted as grinding surfaces to grind the food. Omnivores like us, we have intermediate situation. We still have canine teeth, but our canine teeth in humans are much reduced. In other primates, the canines may be large. We have relatively strong incisors. We use our incisors for cutting food, cutting and tearing. And we have quite strong molars, but not nearly as highly adapted for grinding as they are. But they are not adapted for cutting as they are in carnivores. The really significant adaptations can be seen in the structure of the digestive tract itself. Now, in the case of carnivores, and to a large extent in the case of omnivores as well, there is the major the major function of the digestive tract is to digest things which are relatively easy to digest. In a carnivore, they're eating meat, and that is easily broken down by the digestive enzymes into small amino acids, which are absorbed. And so what you want is actually a gut of kind of intermediate length, which will produce the digestive enzymes to break the meat down. They tend to have a large, small intestine for that reason, and a relatively small colon compared to the small intestine. That is typical for us as well. We have quite a long small intestine, a relatively short colon. And that's because we do actually, in our natural state, depend to a tremendous extent on meat, which is relatively easy to digest and relatively easy to absorb. In the herbivores, they have a tremendous problem because much of their food is composed of a substance, which is cellulose, that vertebrates simply cannot digest. They cannot break it down. It is a rich source of nutrient because it's composed of glucose, but it's glucose polymerized into a substance which is extremely difficult to break down. Cellulose, and the vertebrates simply cannot produce enzymes which will break down cellulose. Instead, some other system has to be devised. First of all, to extract whatever nutrient is present inside the cells. That is, grind the food so fine that you tear as many cells apart as possible. Once you've done that, you liberate the cell contents, then those are relatively easily absorbed. So first of all, herbivores tend to chew their food and have adaptations for grinding their food very fine. But the second adaptation is to take the digestive system and create areas where highly specialized forms of digestion can take place. They tend to have rather small intestines, short small intestines. And instead, their large intestines are extremely long, and they are very often elaborated. Have a look here. This is a little portion of the large intestine called the cecum. And it's the right where the small intestine joins the large intestine. We have a cecum as well. If I can get this to go backwards, I'll show you here. We have a cecum as well. There it is there, as our small intestine joins the large intestine. But our cecum is relatively small. Attached to it, by the way, is the appendix, which is part of the immune system. But in herbivores, in many herbivores, first of all, the colon is extremely large, and the cecum is very large as well. Those elaborations are so that the herbivore can maintain colonies of bacteria, especially, and other microorganisms, which can digest the cellulose. They maintain populations of microorganisms in the cecum and in the large intestine, which break down cellulose and contribute that to the animal. This takes very elaborate forms in many of them. This is just a simple version of the kind of adaptation that you might see. Here is the structure of the digestive tract of the cow and of associated animals, which we call the ruminants. And they depend, of course, entirely upon plant matter for their nutrition. And they have several adaptations which extract the maximum amount of nutrition from that. They do have to eat a tremendous amount. And what happens is that they will feed and will take in as much as they possibly can and store that food in a first stomach. Here, the stomach is elaborated on. There is a first stomach here, which is called the rumen. And food is stored there and is then passed into a stomach here, a second stomach, which is called the omasum. And it is stored there for quite a long time. And periodically, especially at night or when the animals are at rest, they will pass up lumps of this chewed grass, and they will re-chew it. That is what we mean when you say that the cows are chewing the cud. That is what this is. The cud is pre-digested, pre-chewed food, which is passed back to the mouth for re-chewing. And once it is re-chewed, it is then passed into the rest of the digestive tract. This is the third stomach, which is called the omasum, and is then passed into the rest of the intestine. Why? Why do this? And the answer is that in each of these compartments, there are separate collections of microorganisms specially adapted to that particular environment. And they are actually digesting the cellulose. They are digesting cellulose, and when they do so, they accumulate bulk. They accumulate biomass. And that biomass itself can actually then be digested. So what they're doing is they're growing up microorganisms here, and they are then passing those microorganisms into the rest of the digestive system where they can be digested. In addition, these microorganisms themselves produce things like fatty acids and things like that, which can be absorbed by the organism. As a result, this double chewing and then passing the food through all of these different chambers, the ruminants are able to extract a tremendous amount of nutrient from this really poor food source that they live on. Now, not all herbivores have that same ruminant system, but almost all of the herbivores have some association with microorganisms to do their digestion. And horses, for example, have a very, very long, large intestine in which they maintain the collections of microorganisms that do the digestion. Rodents, many rodents like rabbits, they will actually also chew their food twice. What they do is the food has one pass through the digestive system, and they produce what are called pseudo feces. They're not feces. It's half digested food, which they eat again and pass again. And that is, so they re-chew it and re-extract as much as they possibly can. So there are lots of these different adaptations. Many of the herbivores have to have elaborate adaptation to that diet. Being a carnivore and being an omnivore is easy because we get such a tremendous amount of nutrition from meat, which is easily digested. Now this microbiome, we need to say a word about the microbiome, especially as it is now, the human microbiome is now a subject of a tremendous amount of interest. And the interest in the microbiome has been sparked by the fact that we now have ways of assessing how many species of bacterium are present, even when we're not able to culture those bacteria. And it turns out that many of our gut bacteria are in fact completely unknown, but we can see that they are there by looking at their DNA sequence. And it turns out that, for example, our intestine harbors more than 1,000 species of bacteria. Of those, only about 400 have actually been identified. But when we look at the whole digestive system, we harbor about 40 trillion bacterial cells. We have more bacterial cells in our gut than we have cells in the rest of our body. And these, I've already told you, that we know that they contribute very important essential vitamins, the whole B family, for example, and vitamin K. They also provide fatty acids and probably other nutrients as well that we don't even know about. But what is so interesting about it is that they interact with both the endocrine system and the nervous system. And they seem to have a very strong influence on our immune system as well. So our gut microbiome, this is not just a foreign collection of bacteria, they're actually part of us. And our interaction with them is actually regulatory. And the health of our gut biome is actually often a reflection of the health of the rest of the organism. I'll just show you quickly. There are some special ones that we should talk about, some special situations. So first of all, here's a really interesting one. This is called Helicobacter pylori. Here it is. This graph refers to this as well. And Helicobacter pylori is adapted to live in the stomach. And it has some really spectacular adaptations. First of all, it has to survive that incredible acid. Very few bacteria can survive the acid of the stomach. The acid of the stomach is actually protective. It kills off pathogens before they can enter the rest of the gut. But Helicobacter pylori has adaptations to survive the acid. Not only that, it seems to eliminate other microorganisms from the stomach. When you compare people who have Helicobacter pylori to people who do not, you see that the people who don't have Helicobacter pylori in their stomachs have quite a rich microflora of other species. Once the Helicobacter arrives, it appears that it eliminates those. But I should also say that Helicobacter can cause stomach ulcers. It doesn't always, but it is the cause of stomach ulcers. And so this is extremely interesting. It might be encouraging a rich stomach microflora. Getting that back might actually be preventative. Here is another one. And this is a situation which arises especially in elderly people, but in anybody who has had very, very rigorous antibiotic treatment. The microbiota of the gut can be entirely wiped out. And once that happens, the gut becomes susceptible to infection by pathogens, and especially by this pathogen here, which is called Clostridium difficile. Clostridium difficile is a huge problem in hospitals. It is quite widespread, and it can be lethal. It causes a very, very dangerous inflammation of the gut wall, diarrhea, and can cause perforation of the gut wall and death. And very, very difficult to treat indeed, until they discovered one thing. And that was that you could have a transplant of gut bacteria from a healthy individual, put that into somebody suffering from Clostridium difficile, and within days, they recover from the Clostridium difficile infection. So the question is then, how do we introduce the microbiome of a healthy individual into somebody's intestine? And the answer is you do a fecal transplant. You simply take feces from a healthy individual, especially somebody who has not had antibiotics, and simply transplant that into the upper part of the colon of somebody, and the bacteria will happily colonize the new gut. Okay, that's just a little quiz. So all of the digestive functions and our absorptive functions, and of course our metabolic functions, are regulated by continual relay of information from the organs to the other organs to the rest of the body. And feedback circuits are the signal feature of this information relay. Feedback circuits actually regulate digestion, they regulate the storage of energy, and they even regulate our sense of appetite and our sense of how much we should eat. So I'm not going to, you don't have to know all the details here. But all I want to show you in this diagram is, first of all, an organ like the stomach can talk to itself through feedback information. For example, our stomachs can gauge how stretched they are, how full they are. And once the stomach is full, it can signal to the stomach walls to begin to produce hydrochloric acid, pepsinogen, et cetera, et cetera. But that signaling in itself can begin to signal to the rest of the body various pieces of information, and can actually get the rest of the digestive system to begin to act in relay, so that it orders the digestive process. Many of these processes actually occur because information is sent to the brain, especially to the area called the hypothalamus, which then sends hormonal instructions to the rest of the body to regulate these processes. So just remember that all of these organs that we've been talking about, they chatter to one another the whole time. They do so by hormones. And we'll hear again about some of them when we talk about the endocrine system. One set of regulations that we do need to talk about, though, in more detail, is how we store energy. Because we take in energy mostly in the form of carbohydrate. And we take it in especially as monosaccharides, the most important of which is glucose. And when we take in a huge amount of glucose, we actually can't use it all. It's a waste to simply void that. And so the glucose is modified, it's put into a long-term storage product. And in our case, in human beings, most vertebrates, that is the form that it is put into is glycogen. Glycogen is a kind of an intermediate form of storage form, largely in the liver and in the muscles. For extremely long-term storage, we actually put we transform material into fats, and it is stored in special cells called the adipose cells. But that is rather difficult to access. We normally access, first of all, our circulatory glucose, then our glycogen, and then the stored fats. But the body has to regulate where these substances go very, very carefully. Circulating glucose in the blood, excess circulating glucose, is actually extremely dangerous. And so the body has to regulate its passage into intermediate storage and into long-term storage. And this is largely the responsibility of the pancreas. And the pancreas is a very complex organ with many different responsibilities. It is both an exocrine gland, that is, it produces substance digestive fluids, such as we've heard of, that go into the lumen of the intestine. But it's also an endocrine gland. It produces hormones, which are regulatory. And there are two hormones which regulate blood sugar, which monitor and regulate blood sugar. And these are, one you've heard of, I'm sure, is insulin. The other one is called glucagon. And what insulin does is it causes cells, all the cells of the body, to take up glucose. When glucose is in high concentration in the body fluids, insulin is released and causes cells of the body to take in the glucose, so that the blood sugar, the blood glucose level drops. When the blood glucose level drops to low, then this hormone called glucagon is produced. And glucagon causes the breakdown of glycogen inside cells. Glucose, which is taken into cells, is converted to glycogen, especially in the liver and in the muscles. But glucagon causes those cells to break down glycogen and release it as glucose to raise the blood sugar level. So by playing these two against one another, we can set up a feedback system which maintains blood glucose level at the optimum. It maintains homeostasis by balancing the two. So these, by the way, the glucagon, insulin and glucagon, are produced in little collections of cells called the islets of Langerhans in the pancreas. And there are two kinds of cells, the alpha cells, which produce glucagon, and beta cells, which produce the insulin. So that is exactly how the pancreas regulates. The blood glucose is monitored by the pancreas itself. And as the blood glucose level rises, for instance, after a meal, the pancreas begins to secrete insulin from the beta cells. Those beta cells cause the transport of glucose from the body fluids, especially from serum, from the blood, into cells, especially of the liver, but all the other body tissues as well. And the blood glucose level should drop really rapidly. And it falls to a normal level between 70 and 110 milligrams of glucose per 100 milliliters. If you're active, and especially if you haven't fed recently or whatever, your blood glucose level might begin to drop. If it drops below the safe level, then the pancreas is signaled to begin to produce glucagon. And glucagon causes the breakdown of glycogen and release of glucose, and the blood glucose level begins to rise. Rise too high, insulin is produced. Fall too low, glucagon is produced. And so the two interact with one another to produce a normal, stable blood glucose level in a typical feedback system. Let me see if I have anything else to tell you here. No. Okay, so that, of course, brings us to the topic of disorders of these regulatory systems. And the one which you must surely have heard of is diabetes. Diabetes, the proper name is diabetes mellitus. And that melia is honey. in Latin. And that in the old days, they diagnosed diabetes mellitus by tasting a patient's urine. And if the urine was sweet, as if it had been sweetened by honey, then that was the diagnostic feature. And that, of course, is because people suffering from diabetes mellitus have very high blood glucose levels. It is the body makes a desperate attempt to excrete the glucose through the kidneys, which is very damaging to the kidneys, and the urine accumulates sugar. And that just could still be a way that you might initially diagnose the presence of the disorder. Now there are two kinds of diabetes. The first kind is type 1 diabetes. And type 1 diabetes arises because of a defect of the beta cells. There can be various reasons why that defect arises. But in many cases, it is an autoimmune, the patient's own immune system attacks the beta cells, and they stop being able to produce insulin. So without insulin, the blood glucose level begins to rise uncontrollably. That is extremely damaging. It is especially damaging to certain tissues. Blood vessels are one, blood vessels in the eye in particular, nerves, and the brain as well. And if the blood glucose level rises high enough, it actually is a crisis situation and the person can actually die. And unfortunately, in the old days, that was actually the case. Most people with diabetes mellitus developed it in childhood. The exact reasons are not known, but they would develop it in childhood and probably wouldn't live very long, maybe into their early 20s before they died. Now, since we acquired the ability to get insulin first extracted, by the way, from pig pancreas, but then later nowadays synthesized. Now people can actually administer insulin to themselves. They monitor their blood glucose, as this person is doing here. They prick their finger and they're going to measure their blood glucose. And if their blood glucose is too high, they will take insulin in the form of an injection and thus lower their blood glucose levels. So that is type 1. Type 2 diabetes is different. It doesn't involve a defect in the pancreas at all. It involves an inability of cells of the body to respond to insulin. Normally, when insulin is present, glucose transporters in the cell membrane become active and transport glucose into the cytoplasm of cells, where it's converted into glycogen. But in type 2 diabetes, those glucose transporters do not respond. And so the blood glucose levels cannot be dropped. And people with type 2 diabetes suffer many of the same effects as type 1 diabetes, and it can be extremely dangerous. But type 2 is more associated with dietary issues. Very often people who are obese, for example, acquired type 2 diabetes, and it's sometimes referred to as acquired diabetes as well. Not always, it's not always that simple. But type 2 diabetes does not respond to insulin, for example, because the cells can't respond to insulin. But it is treated usually with dietary change, exercise and dietary change. Okay, so I think that I'm going to stop there, and then I'm just going to finish this last little bit when I see you again, because I see that we're just about out of time. Okay, there's not much more on this to do. Please remember that next Wednesday you have a quiz, okay, and that is 15 questions, and it's from material that has not been examined. In other words, from your last test. Please, these quizzes are also important. They contribute significantly to your grade and will form part of my assessment for your midterm assessments as well. Okay, then I'll see you then on Monday.